Now I'd like to welcome today's presenter, Kristen Sainani. Kristen is an associate professor at Stanford University. She teaches statistics and writing, works on statistical projects in sports medicine, and writes about health, science, and statistics for a range of audiences. She authored the health column Body News for Allure magazine for a decade, and she is also the statistical editor for the journal Physical Medicine and Rehabilitation, and she authors a statistics column, Statistically Speaking, for this journal. She teaches the popular massive open online course, Writing in the Sciences, on Coursera. She was the recipient of the 2018 Biosciences Award for Excellence in Graduate Teaching at Stanford University.
Hi, so today I'm going to be talking about how to be a statistical detective. And you might be wondering why you'd want to be a statistical detective. Why is this important? So unfortunately, there are a lot of statistical errors in the medical literature. When people do surveys of the literature, they find that a high percentage of papers have at least one error in either the statistical analysis or in statistical reporting. And I'm showing you just some of those examples here. You can see that it's really a high percentage of papers. In fact, in one survey, they found that just that 19% of the papers they reviewed had just simple numerical mistakes. Now, these types of errors should never happen, and yet they are alarmingly common. So today, I'm going to share with you some tools and tricks that you can use to catch errors in your own work before you submit things for publication or when you're doing peer review. Now, I spend a lot of time doing statistical detective work because I do statistical review for journals. I also review papers post-publication. So, for example, sometimes a journalist might ask me to vet the statistics in a paper prior to them writing about it. And interestingly, the vast majority of errors that I catch are not sophisticated. They are not subtle problems in a fancy statistical model. These are much more basic things. It's things like, the authors added wrong. You can catch most of these errors even if you don't have an advanced degree in statistics. What it really takes is just common sense, the ability to add, subtract, multiply, and divide, and some really cool, easy to use online tools. And I'm going to show you three of my favorites today: Statcheck, Brim, and WebPlotDigitizer. And then finally, of course, it's also helpful to have a good understanding of some basic statistical concepts. So today I'm going to walk you through three case studies that illustrate the use of these tools. The first case study dates back to 2006. There was a paper in Nature Medicine that garnered a lot of attention. And the authors claimed that they could use publicly available data to identify genetic signatures that would predict a cell's sensitivity to specific cancer drugs. And the idea was that you could use this then to match cancer patients to the right drug for their tumor. And this was named actually one of the top 6 genetic stories of 2006 by Discover magazine. And there was a lot of interest around this. And so there were some physicians at MD Anderson who, because this was based on publicly available data, wanted to institute this or implement this in their own institution. And so they approached 2 MD Anderson statisticians, Keith Baggerly and Kevin Coombs, and they asked for their help in recreating this method. When Kevin Baggerly and Keith Coombs started digging into the method and the data, however, they started to uncover a number of problems. And I'm just going to share one example with you. So this method is based on publicly available data. Some of the original data were published in The Lancet in 2003. And what you're looking at here are breast cancer cell lines. The blue cell lines are sensitive to the chemotherapy drug doxycycline, whereas the red cell lines are resistant to the drug. The authors of the Nature Medicine paper used the exact same data, and they claimed that they had come up with a genetic signature that could separate the resistant cells from the sensitive ones. Now, what I want you to do here is actually just take a careful look at the top panel versus the bottom panel and see if you can spot a problem. And this is basically like that spot the difference game that you played when you were a kid. So look carefully at the top panel and the bottom panel and see if you can spot the problem, if you can spot the difference. And keep in mind that blue in both graphs means sensitive and red in both graphs means resistant. If you're looking very carefully, you might notice that in the top graph, there are 11 sensitive cell lines and 13 resistant cell lines. But in the bottom graph, these have been exactly reversed. There are 13 sensitive and 11 resistant. What happened is that the authors of the Nature Medicine paper got this wrong. They mixed up the resistant and sensitive cells. The reason this happens is that we often code binary variables as ones and zeros. So in this case, maybe 1 meant sensitive and 0 meant resistant. But when the authors of the Nature Medicine paper reanalyzed the data, they got it backwards. They mixed it up, and they thought that 1 meant resistant and 0 meant sensitive. This is a big problem because it turns out they ended up using this algorithm in clinical trials at Duke University to treat cancer patients. It turns out that this algorithm was complete bunk. It was not at all predictive, but had it been predictive, it would have actually been assigning cancer patients the exact wrong drug for their tumor. Now, this is not a sophisticated error, right? This is just messing up what 1 means and what 0 means. But this can have profound consequences, and it's the type of error that really worries me. This story, this example, is actually part of a larger backstory. It turns out that Baggerly and Coombs uncovered a whole bunch of data manipulation errors, reversals of 1s and 0s, off by 1 errors in Excel, probably fraud as well. And so it ended up that this Nature Medicine paper eventually got retracted along with a host of other papers by the same research group. The clinical trials at Duke were halted, there were lawsuits, so this was actually part of a larger scandal. All right, the second case study comes from a paper that I was asked to review by a journalist. And this paper was trying to get at this idea that sometimes when people exercise more, they don't actually lose weight. And one of the reasons is that sometimes if you exercise more, you end up compensating by rewarding yourself with more treats. And so this paper, this study was trying to get at that. So they randomly assigned 56 participants to either an exercise condition or a fun condition. The exercise framing group, there were 28 of them, walked a mile, and they were told they were walking for exercise. Whereas the fun framing group, they also walked a mile, but they were told that the experiment was actually about music, not about exercise. So they were given an MP3 player and they had to rate some songs. After the study was over, all of the participants got a buffet lunch. And the researchers surreptitiously measured the number of calories that each participant served themselves during the lunch. This table shows the main results from the study. I'm just going to zero in on a few items from this table. So first of all, they were comparing 2 groups, the exercise framing group and the fun framing group. So those are the rows of the table. The columns of the table correspond to different calorie counts. So the first column is just the total calories they served themselves at lunch. They then broke this down to calories from the main meal versus calories from the drink and the dessert. And they had 2 choices on dessert. They could have chocolate pudding or applesauce, and the drink was either a Coke or a water. Now, the first finding that the authors report actually is that there was no difference in the groups in the total calories served. The average calories in the exercise group was 313 versus 305 in the fun framing group. So virtually no difference there. However, the authors really wanted to find a difference between the groups, so they kept on digging and looking at different components of the meal. And so when they looked at the drink and dessert calories, what they called the hedonic choices, they actually found a difference between the groups. So the average calories from drink and dessert was 133.98 in the exercise framing group versus 94.45 in the fun framing group. And they claimed that this was a statistically significant difference, and they actually made a big story out of this finding. And largely the story of their paper was built around this result. Their take-home message was, see, if you tell people that they're exercising for exercise, they end up eating more sweets afterwards. Now, you might notice right away that this is not a huge difference, right? This is a 40-calorie difference, so it's not a really big difference. Even more than that, if you start digging into the table, you start to notice some numerical anomalies. First of all, there are some curious patterns of missing data. If you look at the exercise framing group, in all of the columns, n equals 28, meaning that there was no missing data. All of the participants were included in the averages for those variables. However, if you look at the fun framing group, for the total meal served, there was no missing data. There was 28 people. But then if you look at the regular meal, the main meal, and the drink and dessert, the n is 24. So we have some missing data. And this pattern is a bit curious to me because if you have the calories, the total calories for someone, that would imply that you had measured the calories on the individual components of the meal. Now, of course, there could be some innocent explanation here. Maybe somebody forgot to write down where those calories came from, so they had the total, but not the individual components. There could be an innocent explanation, but it does raise a red flag for me. It makes me wonder if maybe they threw out a few data points that they just didn't like in order to get the result they wanted. There's also a whole bunch of numerical inconsistencies if you start looking carefully in the table. So for example, the total meal consists of the regular meal plus the hedonic choices, the drink and the dessert. In the exercise framing group, as I said before, there was no missing data. So what this means is that the average calories from the regular meal plus the average hedonic calories has to add up to the average calories from the total meal. But when we do a quick check of this, when I add up the 193.67 plus the 133.98, I get 327.65. The latter two columns here should add up to the first column, but they don't. 313 is not equal to 327. I don't know why these don't add up, but they should. We see a number of other problems like this if you keep digging in the table. So for example, the hedonic choices consist of the dessert plus the applesauce. So the latter three columns here should add up to the first column. The pudding plus the applesauce plus the drink should equal the overall hedonic calories. Now this calculation is slightly more complicated because if you look carefully, what you'll notice is that when the authors reported, for example, the chocolate pudding served, they didn't report for all 28 people. They just reported on average among the 11 people who took pudding, how much pudding on average did they take. So in order to calculate the average hedonic calories per person here, we have to do a little bit more work. We have to first add up what was the total hedonic calories for the group. So the total amount of calories taken from chocolate pudding, applesauce, or drink, and then divide by 28. So for example, I multiplied 135.3 times 11 to get the total chocolate pudding calories taken in this group, plus 75.14 times 16, plus 33.1 times 29. That gives me the total calories. I divide by 28. I get 130.37. Now, that is supposed to equal the first column here, the 133.98, but it doesn't. It's just a little off. So something funny is going on here. I then took this paper and I ran it through an online statistical checking tool called Statcheck. And this is a great tool because it's super easy to use and it does some text mining for you. Now, it won't work on all papers. The statistics have to be presented in the text in a certain way, but the fun and exercise paper, you can use Statcheck on that paper. So what it does is it does text mining and it automatically extracts test statistics, degrees of freedom, and p-values from the text. And it simply checks those to make sure that they're internally consistent. So for example, let's say you have a paper where you're comparing two groups and you run a t-test. And you get a t-statistic out of 1.89 and your degrees of freedom is 19. The degrees of freedom is related to your sample size. A t-statistic of 1.89 with a degrees of freedom of 19, the p-value associated with that, the correct p-value is 0.074. If a paper reports that t-statistic but reports a p-value of 0.045, then something is wrong. Those are inconsistent. And Statcheck automatically flags those for you. Statcheck is implemented in R, but it's also implemented in this convenient web app that doesn't require you to know any programming at all. And it takes about 1 second to use this. All I did here was you just upload your paper. So I uploaded a PDF of the fun versus exercise article, and then it runs in a second, scans the whole article, extracts the statistics from the text, and checks them for consistency. So I'm showing you here the result that I got. There's actually more. I've cut the screen a little bit. There's more results that it flagged, but it flagged a number of the statistics from the text as inconsistent. And so I'm going to just show you 2 examples of the statistics that it flagged as inconsistent. So first of all, one of the things it pulled out of the text was this F statistic of 2.791. The authors had reported that that corresponded to a p-value of less than 0.05, which would be considered statistically significant at the 0.05 level. Statcheck says, wait, that's supposed to be, that F statistic is supposed to give a p-value of 0.101, so something isn't matching here. Well, I dug into the paper and found out in the text where did that 2.791 F statistic come from? And it actually corresponded to a comparison that we've already seen today, which is of these hedonic calories, the 133.98 hedonic calories in the exercise group versus the 94.45 in the fun group. And this is that result that the authors made a big story of. So when they made that formal statistical comparison, they got this F statistic of 2.79. They then reported, though, P is less than 0.05, and then they've slipped in something in the text. They don't put this in the table, but they slipped in the text here, this little bit of a sleight of hand. They say, actually, we're not reporting a two-tailed p-value here. We're reporting a one-tailed p-value. Now, there's actually no reason to report a one-tailed p-value here. And in fact, in most of the p-values they report in their paper, they report the two-tailed p-value. The reason they are not matching the p-value from Statcheck, however, is the one-tailed p-value is half the two-tailed p-value. So the correct p-value really is 0.10104. But what they did is said, well, we really want to get that p-value below 0.05. So we're going to, instead of reporting the two-tailed p-value, we're going to report the one-tailed p-value. And then they've done a little trickery with somehow rounding down because, of course, half of 0.10 should be equal to 0.05, but they're somehow saying that that's less than 0.05. This is bad statistical practice. Making statistical choices just to get your p-value to be less than 0.05, that's something we call p-hacking, and that's bad science. Now, there's one other example that they flagged that I'm going to zero in on here. So there was another F statistic of 2.83. The authors of the paper reported that the p-value was 0.04. Statcheck says, nope, that p-value should be 0.09829. So I again dug into the paper to see where this statistic came up in the paper. And it corresponded to another comparison that the authors made. They asked the participants to rate their how exciting they found the walking activity on a scale from 1 to 9, where 9 means extremely exciting. And they found that the fun group rated the excitement as an average of 7.19, which was a bit higher than the exercise group, which rated it at 6.45. When they made a formal statistical comparison between those two means, that's where that F statistic of 2.83 came out. But notice they're making the same little trick as before. They report the p-value as 0.04 because they were reporting the one-tailed p-value. Again, there's really no reason to report the one-tailed p-value here. They're just doing it so they can claim p is less than 0.05. All right, I then took that same paper and ran it through another one of my favorite online statistical checking tools. This is a tool created by James Heathers and Nick Brown. And what it does is it flags impossible mean values. So let me just give you an example. Imagine you've got a variable that you've measured on an integer scale. So maybe you asked your participants to rate their pain on a 0 to 10 integer scale. And imagine also that you have a small sample. So maybe you have a sample size of just 10. If you have an integer value and a sample size of just 10, there are only certain mean values that are possible. If you sum up 10 integers and divide by 10, then the mean has to contain an even 10. So you could get a mean of 4.00 or 4.10, but you cannot get a mean of 4.01 or 4.15. So if the authors report a mean of 4.15, then you know something is not quite right. This has been implemented in both a web app as well as a phone app. It's very easy to use. All you have to do is take your mean and your sample size from your paper, enter it into Grim, and it's going to tell you whether or not that mean is a possible mean. So I went ahead and I entered that 133.98 average hedonic calories from the exercise group. The N there was 28. I entered that into Grim. I tried various ways of rounding, and in all cases, it tells me that 133.98 is inconsistent, meaning that is not a possible mean if calories were measured as integers and I have a sample size of 28. Now, there is a possible explanation here. We normally report calories in integers. Like if you look at calorie counts on a food label, they're always going to be in integer values. It is possible that when the researchers here recorded the calories into the database, they recorded calories out to 1 or more decimal places. That could, therefore, cause the mean to be 133.98. So that's possible. However, remember, we've already looked at something in this paper that we know for sure was measured on an integer scale. So the researchers asked the participants to rate how exciting they found the activity on this Likert scale from 1 to 9. And this is definitely integer values. And remember, the result here was that the fun condition had a mean of 7.19, and the exercise condition had a mean of 6.45. And I can tell from the degrees of freedom in that F statistic that there was no missing data used in calculating those means. All 56 participants were used in that comparison, meaning all 28 per group were used in calculating those means. So I can run this through the GRIM checker. And when I do that, I put in 7.19 and sample size of 28. Lo and behold, it tells me 7.19 is inconsistent. It's not a possible mean. And again, I tried all versions of rounding. GRIM allows you to try different versions of rounding. It's inconsistent in all cases. Same with when I put the 6.45 and 28 into GRIM, also tells me, nope, this mean is also inconsistent. So this paper just has a lot of red flags in it. And it actually has a backstory as well. So the reason that I was looking at this paper for the journalist is because one of the authors of the paper has actually had 18 other papers retracted. Now, this particular paper has not been retracted, but the author has had a large number of papers retracted due to the same kinds of problems: numerical inconsistencies, sloppy data handling, p-hacking. So it might not be surprising that we're seeing so many problems in this particular paper. It was actually the inventors of the GRIM test, Nick Brown and James Heathers, who actually uncovered a lot of the problems in those papers that led to their retractions. All right, so the first two case studies I showed you were actually tied to a larger scandal of scientific misconduct. I just want to show you that the majority of errors that you catch are not tied to some larger scandal. So this third case study is just a study I happened to come across. There's one finding in the study that I think is not robust, so I'm not criticizing anything else in this study, and it is not tied to a larger scandal. But this was a study that happened to get some press attention last year. It was a study on breastfeeding and stress in babies. So it's well known that breastfeeding reduces stress in babies. And the claim of this paper was that the authors had found a genetic mechanism that explained how breastfeeding might lead to lower stress in the babies. And there were two main findings. So the first finding was they looked at 41 mother-infant pairs, and among the babies that were breastfed, they found lower DNA methylation of two genes related to stress. And DNA methylation is a way of regulating gene activity. That was the first finding. The second finding was that they then correlated this lower DNA methylation of these genes with lower stress in the babies, lower stress reactivity. And it's just that second finding that I'm going to zoom in on now. So the authors actually published a plot in their data that shows the correlation between DNA methylation of the gene and low and higher stress reactivity. And so what you're looking at here, the x-axis is the amount of DNA methylation of the glucocorticoid receptor gene, which is a gene involved in stress. The y-axis is cortisol reactivity, which is a measure of the stress in the babies. The authors reported a correlation coefficient here of 0.41, which would be considered weak to moderate correlation, and it was statistically significant. If you look at that plot, it does look like, yeah, it seems like as the methylation goes up, the stress reactivity goes up. It looks like there's some association here. It's important to understand, though, that when you superimpose a straight line on a graph like this, it can be misleading to the eye. And so what I did was just to pop this particular graph into just Microsoft Paint, and I sort of crudely erased that line. And when you look at the graph without the line, you get a different impression. So let me go back. Here it is with the line. The line is kind of drawing your eye in a certain direction. When you take the line off, what becomes apparent is that association doesn't look so strong anymore. What we really have is just kind of this one cloud of points down here. And in that cloud of points, it doesn't look like higher methylation really correlates to higher stress. But then we have this one data point up here. We have this one baby who happens to have both high methylation and high stress reactivity. And the concern here is that that one point might be what we call an influential data point. That one point might be drawing the line toward it and causing us to see an association that's really not robust. And so I wanted to probe these data further. And I used a tool, one of my favorite online tools, called I'm reviewing papers, because most times when we're looking at papers, the authors have not provided the underlying raw data. I wish they would, but in a lot of cases they haven't. WebPlotDigitizer gives you a way to extract that underlying raw data from plots or images in the paper. And so, for example, if you have a scatter plot like this, each of these data points corresponds to a methylation level and a stress reaction level. that XY coordinate is what WebPlotDigitizer can extract. So WebPlotDigitizer extracts the XY coordinates of each one of these points, thereby recreating the underlying data set, giving you back those methylation levels and those stress levels. It puts it in a CSV file that you can then download for further analysis. So this is super cool and useful. It's quite easy to use. It's implemented in an online app. I was able to download these data from WebPlotDigitizer in about under a minute, and I have a little demo video that just shows you how easy it is to use this particular tool.
To use WebPlotDigitizer, go to their website, upload the image that you want to extract data from. Then you have to give some information about the scale of the axes. So you click on 2 points on the x-axis and 2 points on the y-axis and enter those coordinates. And now you're ready for data extraction. To use the manual data extraction, you simply click on all of the data points that you see on the screen. And once you've hit all of them, you then hit View Data. And now you have a data set that's ready for download into a CSV file that can be used for further analysis.
So you can see that it's super easy to use. And what I did was I took that data set, I extracted the data in WebPlotDigitizer. Again, it took me less than a minute. And you don't need to know any coding or anything, super easy to use. And then I just verified that I was able to reproduce the original analyses that appeared in the paper. So I popped my data into SAS. What you see on the right here is my reanalysis. I'm able to regenerate the same basic plot, and I also get an R, a correlation coefficient of 0.41, exactly what the authors reported. My p-value is 0.01. They reported p is less than 0.05. So it's a good match. This just verifies that I extracted the data correctly. Once I know that I've extracted the data correctly, then I can do some reanalysis. So I did 3 things. So first, I've already told you that a straight line can be misleading to the eye. So I removed that straight line and I superimposed instead what's called a Loess line. And this allows a little bit of curvature and can better reveal the underlying pattern in your data. I also calculated what's called a Spearman correlation coefficient rather than our standard Pearson's correlation coefficient. The Spearman correlation coefficient is based on ranks rather than absolute values, and therefore it's less susceptible to influential points. And then finally, I repeated all of the analyses, removing that potentially influential point so I could gauge just how much influence that one point was having on our analysis. All right, so when I kept that influential point in, you can see this is just the original data set. Same data set, I haven't changed anything. And I just superimposed that Loess line rather than the straight line. And you can see that that Loess line sort of shows you, yeah, there really isn't much going on for most of the data points, but that line is being drawn by that one point upwards. The Spearman correlation coefficient here turned out to be much lower than the Pearson. So it was 0.41, and now we're seeing a correlation of only 0.18, which would be considered very, very weak correlation. It's no longer statistically significant. If I remove that influential point, what you'll notice is there's basically no association left. We have now mostly a flat line. Both of the correlation coefficients are about 0.1, which would be considered minimal correlation. The p-values are very high. In other words, there doesn't appear to be a lot going on here once we remove that one influential point. That entire association was really driven by that one data point, that one baby who happened to have both high methylation and high stress reactivity. Now, it could be that if we were to sample more babies in sort of that area of the graph with very high methylation, it might be that those babies would also have high stress reactivity and that this association would hold. But as it stands now, we are basing the entire association on one data point. And it could also be the case that it just happens to be by chance that that baby happens to have both high methylation and high stress, but that those two things are not associated. So if I were reviewing this paper, what I would have told the authors to do was to show some of these additional analyses and also to moderate their conclusions accordingly. All right, so my take-home messages for today are statistical errors are common in medical studies. Now, some of these errors may have no consequences. They may be very minor, but others can completely undermine a study's conclusions. A lot of these errors don't take an advanced degree in statistics to catch. And I've given you some tools that you can use to catch these errors. Of course, we would rather prevent these errors from occurring in the first place rather than have to catch them after the fact. And so the way that you prevent errors is to follow best practices in statistics, principles of effective statistics. And these are the kinds of things that I teach in my course. So for example, if you're going to be analyzing data, the first thing that you need to do is to check and understand your data. You need to know what 1 stands for and what 0 stands for, and you should double check that and triple check that. In fact, most of a data analysis is spent in this step, checking and understanding your data. And one of the common errors I see when students come to me or researchers come to me with data, they come into my office and they've got some output from a statistical analysis program. And they say, oh, can you help me understand what this output means? And I say, okay, well, let's go 5 steps back. What have you done to clean your data, to check your data, to graph it, make sure you've checked that there aren't any funny data points? And a lot of times people have skipped that step. They're really, really happy that they were able to get their data into a statistical analysis program and then something came out, yay. And they skip the most important steps of a data analysis, which is where you're merging data, checking data, making new variables, cleaning data, understanding what you have in your data set. And in fact, when I do a data analysis, I spend 90% of my time doing this step. 90% of my code will be about checking and understanding the data and making new variables and so on. So that's really most of what happens in a data analysis. You also want to pay attention to simple numbers. So make sure that your ends add up and that simple numbers in your paper add up. Draw lots of pictures with your data. It's always easier to understand what's going on with your data if you graph it. Use common sense. It's a funny thing about statistics that often when people start doing statistics, I think they're so intimidated by it that they throw common sense out the window. So just go back to basic common sense. Consider the totality of the evidence. So don't just cherry pick the 1 or 2 tiny results that seem to support the story that you want to tell. Really understand what the data are trying to tell you. Keep an accurate record of all your data manipulation and data analysis in code. Please, please, please do not manipulate data or analyze data in Excel. It's very easy to introduce errors in Excel. That's actually what happened in that case study with the Nature Medicine paper that I told you about earlier. You want to port your data set into a proper statistical analysis program like SAS or R or Stata, and anything you do to the data, merging the data, making new variables, you want to do all of that in code and keep an accurate record. This actually helps you to prevent errors. It also helps you to trace back an error if an error ever occurs. Finally, if you're going to be analyzing data, it's important that you understand the basics of probability and statistical inference. This doesn't mean that you have to be a whiz in linear algebra or calculus. It does mean, though, that if you're going to be analyzing data, you ought to understand some very basic concepts like what is standard deviation and what is standard error. It's a pet peeve of mine that there are a lot of people running around analyzing data that don't know what standard error is, even though that's like a column that comes out in every single one of your statistical outputs. So understanding those basic concepts is really important in being able to get things right and interpret things correctly. So to help you to do better statistics and to follow principles of effective statistics, I have a few resources I want to point out. So I've been writing a column for the journal PM&R for over a decade now. There's probably 35 or 40 columns at this link. They cover some basic concepts in statistics, but also some more advanced topics. So like if you want to learn, say, about principal components analysis or instrumental variable analysis, I may have a column on those things. I am also, as of today, launching a new online training program, Medical Statistics 1, 2, and 3. You can earn a certificate in medical statistics if you complete all 3 courses. So I'll just say a few words about those 3 courses. I'm really excited about the new program. So each one of these courses is 4 units long. They'll take you about 15 to 20 hours to complete. And we will be teaching you programming in either SAS or R. You do not need to have any prior knowledge about SAS or R. You can have never opened SAS or R before, and that's just fine. We'll start at step 1. We will be using the free version of SAS University Edition, or you can use R. You do not need to do the courses in both of those. You can just pick one. Of course, you're welcome to do it in both. The course is implemented in both languages. In MedStats 1, you will be learning that introductory programming, descriptive statistics, data visualization, and also principles of effective data analysis. MedStats 2 goes through the basics, the foundations of statistical inference, probability, what is a p-value, what is Bayesian inference, common statistical pitfalls, and also one of my favorite things to teach, which is computer simulation. And just to give you an example of how powerful computer simulation is, there's a classic probability problem called the birthday problem. And it goes like this. If you have 30 people in a classroom and you ask, what's the probability that 2 people are going to share the same birthday? Not the same year, just the same day. It turns out that most people think that's going to be pretty low. And it turns out that that probability is actually 70%, surprisingly high. Now, you can solve that birthday problem with probability, with permutations and combinations. Really fun problem to solve mathematically. I'll show you how to do it, but it's a little complicated. You can also solve that same problem just using computer simulation, which is much easier, much more intuitive, few lines of code. And all you do in a computer simulation is you have the computer randomly select 30 birthdays out of the possible set of 365 birthdays. Imagine you had a big dice that with 365 sides, and you just roll that 30 times, come up with 30 birthdays, and you check to see if there's a match. And then you have the computer do this a large number of times, maybe 100,000 times. Your computer will do it in a few seconds. And you just see how frequently were there matches, how many classrooms out of 100,000 had a match, and that's your probability. It gets you the same answer as the mathematical solution, but it's much easier. It can also solve more complicated problems. So for example, if I ask, what's the probability that 3 people share a birthday? That's hard to solve mathematically, easy to solve on a computer. And so then MedStats 3 will then cover specific statistical tests for different types of data that are common in the medical literature, as well as plots that go with those statistical tests and regression analysis. And so I think I'll open things up now for questions and answers on my talk and on the courses.
Thank you so much, Kristen, for the informative presentation. We'll go ahead and take some Q&A questions now. Continue to submit your questions via the Q&A box there, and I'll address them with Kristen. So the first question here is, for the exercise test, what role does sample size play, if at all, in the validity of the study?
So I think this question is about the fun and exercise paper. Yeah, the sample size is not huge here. That 28 per group. It's not the worst I've seen, but it's not huge. And so obviously, the more people you sample, the more precise your answer is going to be. That is, you're going to have a better sense of the answer. And if you do very small studies, what you can show is that if you have studies what we call underpowered, you can show that when you find in the literature significant results from small studies, those significant results are more likely to be false positives than if you had run a study that was properly powered. So there are really good reasons to make sure that you run properly powered studies, which usually means having a big enough sample size. And that's one of the main concerns with small studies. It's easier to find something just by chance in a small study, and then we tend to, it can be statistically significant. We make a story out of it. Those studies are more likely to turn out to be false positives.
Okay, so the next question, and I believe this might be related to the fund framing activity. Did they at least justify why they were using a one-tailed p-value?
No. So again, this paper has a lot of red flags. I think it's tied to an author who has actually had taken an early retirement from Cornell for these reasons. They did not. In fact, they don't even mention in the table that it's a one-tailed p-value. They just kind of slip this in the text. And there is no justification. Most of the p-values reported are two-tailed, except for the ones that they wanted to get under 0.05, then suddenly it's a one-tailed. There's no explanation anywhere in the text. If I were doing a review of that paper in peer review, I would have told them to explain that choice. And maybe they had an explanation, but my suspicion, again, given the larger context here, is that they were simply trying to get the p-value less than 0.05.
When there is a small sample size and a strong or highly significant p-value is shown, can we rely on this value? And some people state that the results are very strong as there is a highly significant p-value with such a small sample.
Yeah, so one of the problems in the medical literature is we have put too much emphasis on the p-value, and people read too much into a p-value. I'm not saying that p-values are useless or that they're bad. It's just that they are one tiny piece of information along with a whole host of other information that you're supposed to look at. And unfortunately, sometimes people pay too much attention to the p-value. Now, it is true that the smaller the p-value, that's indicating something about your results. And so if the p-value is less than 0.001 versus less than 0.05, you can show, take some other assumptions that you have to work in, you can show some things like it's less likely to be a false positive. So you can have more faith in the idea that maybe that's not a false positive if the p-value is much lower. But there's a lot of factors that go into determining whether something is a false positive. It depends on how well the study was powered and some things like this. So you don't want to just pay attention to that p-value. You want to pay attention to the entire context of the study. Was there a likelihood of bias in the study? How big was the study? Was it well powered? You could get a really big difference between two groups for a number of reasons that don't have to do with the groups actually being different in the real world. So that, yes, if the p-value is less than 0.001, I'm at least going to be less worried about chance fluctuation, but I might still be worried about all these host of other things that could have gone wrong. And you always want to pay attention to the effect size and the study design, and on the larger context, p-value is just that one little piece of information.
The next question is, how do I know the proper sample size for my study?
That's a great question. So before you do a study, you should always do a sample size calculation. It's great to work with a statistician on that to make sure you're doing it correctly. And the idea there is, forces you, before you do a study, not only to get the sample size right, but when you do a sample size calculation, it forces you to figure out, what is the purpose of my study? What is the hypothesis that I'm trying to test? What is my primary outcome and my primary independent variable? If you set those things up ahead of time, you are much less likely to end up doing some bad statistical practices where you're just digging things out of chance fluctuation. So we always want to understand up front, before you do the study, what is the research question? What are your main primary hypotheses? And then you use that information to do a power calculation to make sure that you have a properly powered study. And unfortunately, a lot of times people don't do that, and that can lead to problems.
Okay, so another question is, why do simple statistical errors sometimes pass through the peer review process?
That is a great question. A lot of the errors I showed you today should never have made it through peer review. That fun and exercise paper, I don't know how it made it through peer review. There were so many red flags in that one. I think the peer review system is the best system we've got. I think it's better than not having peer review, but unfortunately it's imperfect. You get a lot of people volunteering their time. They don't necessarily have the time to vet every single number in the paper. And sometimes they don't feel confident in their ability to vet those numbers. And so things get through that shouldn't get through. There are journals who employ statistical reviewers. So I do statistical review for some journals. And as a statistical reviewer, it's nice because I don't have to pay attention to everything about the paper. I can just hone in on those numerical values. That helps me to uncover some of those errors. Whereas if you have to pay attention to everything about the paper, the study design, the wording, how important was the question, then sometimes you lose track of the numbers. So I wish we, did better in peer review, but you can only spend so much time on a paper, and checking every number is often not what reviewers are able to do. It would help if authors provided their underlying raw data, but unfortunately, that's still not common in medical studies.
So besides the tools that you've described today that you mentioned are your favorite, are there other tools that you've encountered?
Sure. So, I use SAS or R a lot because some of the things like the Statchecker to check that things are internally consistent, I can also pop that statistic into SAS or R and check it there. What Statcheck is doing in checking your T statistic and your degrees of freedom to see if it matches the p-value. If your paper that you want to check is not set up in a way that it feeds through Statcheck nicely, there are online calculators that actually you can just plug in the T statistic and it'll tell you the p-value. So I end up using a lot of online calculators. There's also online calculators that take summary data. So you can take the means from a paper and the sample sizes and the standard error, things that are, or the standard deviations, things that are typically given in a table of the paper. You can plug those into these online calculators, like there's online t-tests. And then you can check to see if the author's got a p-value that matches what you get. So there's a lot of online tools that are really useful if you Google around a little bit. And happy to, if you want to follow up and ask more about those, I'm happy to send other links around.
So the next question is, how do we know if we should remove influential points if these points are in fact the important data that reflect abnormal patterns of attention?
Yeah, so that's a great question. So one thing is, a common question I get asked is it okay if I remove this point? I think it's an outlying value. I want to take it out of my data set. So we never just remove data. So first of all, we're not going to throw that data out. What you're going to do, though, is you're going to present two sets of analyses, one that includes that data point and one that doesn't, so that you can show the reader, hey, this data point is having this amount of influence on my results. And then the reader can interpret accordingly. So the example with the baby, that one baby was really the entire story. And like I said, it could be that if you had more babies sampled with that high of methylation, you would see the same thing. But you want to let the reader know this is really due to one data point. And so it's important to actually run two sets of analyses. We call that a sensitivity analysis. You're going to show both sets of results.
Okay, so I'm about to conduct pilot trials for my research and biofeedback. What can be the effective sample size that I can take in to a pilot?
Right, so pilot studies often are hard to know the correct sample size because you are doing the study for reasons other than to show whether or not the outcome is efficacious. Usually a pilot study is done simply to test the procedures of the randomized trial, like that, that your randomization works, that people are going to be randomized correctly, that people will actually enroll and actually finish your study. So it's really more about feasibility and also to get some preliminary data that could then be used for a power calculation for a larger study. So, you can do some kind of power calculation for a pilot study, but often pilot studies are more guided by your logistical constraints, and that's okay because as long as when you finish that pilot study, you're not going to draw any conclusions about the efficacy of the drug or the intervention. Rather, you're going to take that information and use it to do a bigger trial.
Should every study use modeling, generalized mixed models, GEE, etc.?
Okay, that's a good question. So not necessarily. You can have a really great study that is purely descriptive. So I don't mind a study that just gives me means if that's useful information. This is the percentage of people that do X. That might be really useful information. And this is one thing when I get students coming in with data, and sometimes they don't know why they collected that data. I say, maybe what you were after is just something descriptive, and you're not calculating p-values or comparing groups. You just want to have some understanding of, how many, I do a lot of things in athletes, how many women athletes are doing X, Y, or Z. And that's totally fine. So not every study even needs to have what we call inferential statistics, like formal comparisons of groups. If it's a very simple study and a t-test will do, is there a reason to use something more complicated? Well, a t-test is just a specialized case of linear regression, for example. If I'm reviewing a paper and you've used a t-test, will I say you have to use linear regression? No, they're the same test, and one is a little bit simpler to interpret. So if it's appropriate, a simple test is just fine. There are lots of cases, though, when I'm reviewing papers where I will say, a more sophisticated model would have helped to deal with some of the issues in the paper, like missing data, and clustered data. There's lots of cases where those more sophisticated models are actually useful. Time-changing predictors, things like that, can be handled in a more sophisticated model.
How reliable for meta-analytic purposes do you consider the WebDigitizer?
Oh, I know a lot of people who use the WebPlotDigitizer for meta-analysis, actually. So that is one way to get the data if it's only available in a figure. I think it's very reliable. I think there's actually been some studies, I don't know any numbers on, but that WebPlotDigitizer has two modes. You can do it in the automatic mode, and you can do it in the one where you actually have to click on the points. Now, the problem with the automatic mode is it will put dots on extraneous things on your graph. So sometimes you will have to go through manually and remove those. So that you might use the automatic mode in a combination of the manual mode to make sure that you're getting those dots quite right. But I found it to be quite, the fidelity is quite good with the underlying data. And so, yeah, I know a lot of people have used it in meta-analysis.
How can we choose an appropriate correlation test among Pearson, Spearman, etc.?
Right. So Pearson's correlation coefficients have more assumptions. You're assuming that you have normally distributed data. Like I said, they are that statistic is sensitive to influential points. And so if you have skewed data, things that don't follow a nice bell curve or outliers, extreme values, then it's more appropriate to use the Spearman correlation coefficient. And you can usually see that graphically. If you graph the things and it looks like there's a few outlying points or things are very skewed, then you're better off using the Spearman. You can always calculate both, and if they are drastically different, that makes you think usually that's because there is something violating the assumption of the Pearson's correlation coefficient.
So human genome is big data. What are your thoughts about statistically associating genes with clinical symptoms?
Yeah, I mean, it's an interesting question. There's been a lot written about this because we have all this great genomic data. We have to be very careful with genomic data because there are lots and lots of data points. And if you have 30 patients and they each have 10,000 data points from their genome, you have to use statistics that account for this, what we call multiple testing, the fact that you're comparing so many things that you're going to find some things that look different between the two groups. You have to be very stringent on what you consider a difference. So there are techniques for that in genomics. It is easy to find spurious results. And some of the early results, like, for example, with microarray analysis that came out, everybody was correlating microarray gene expression with diseases, and a lot of that turned out to be spurious. We have better methods now. I think there's more quality control on that. We know how to do that better statistically, but you always have to be careful when you're dealing with a small number of patients and a large number of variables, as you would get with genomics.
If we have significant outliers in a small sample size, should we take them out of the statistical analysis? And why did you rerun the analysis without this baby with a high methylation and stress?
Yeah, so I mean, the thing on outliers is we never ever want to throw out data. So like, if you always want to check, go back if you can and make sure it's not a data entry error, because that happens too. But if it's a real data point, it's just something where you have somebody that looks different than the other data points, you're never going to throw it out. So we never throw out data. But what you are going to do is you're going to run two analyses, one with the data point and one without it, so that you can see how much influence is that one data point having. It may be the case that depending on the exact configuration, that one data point may actually not have a lot of influence on the analysis. With the case of that baby was having a lot of influence on the analysis. Most of the association was due to that one baby. So then you have to interpret that with the correct caveats that, hey, we're putting a lot on one data point. But you want to do the analysis both ways. You don't want to only show one of those analyses.
What are some programs or initiatives that the medical field or other fields are implementing to address the reproducibility crisis? And do you know if we have evidence about which initiatives or interventions are more effective?
Yeah, that's a great question. I mean, there's a lot of ideas. So here at Stanford, we have the Meta Research Center with Johnny Anitas and Steve Goodman, who are have a lot of ideas about how you can improve the system. The system needs improvement, and really a lot of these things are system-wide. Some things that people think might work is pre-registering studies, for example. So if before you do your study, you actually put down, what is the hypothesis I'm trying to test? What is the research question? Here's how I came up with the sample size. Here's what my primary outcomes were. Then you're kind of bound to that. And it prevents a lot of these sort of statistical shenanigans that can happen after the fact where people are doing p-hacking and stuff. So pre-registration of studies is one. Publishing your data, making your data publicly available. It's an issue in medical studies because we worry about privacy, but there are ways to de-identify data and make that publicly available. That would go a huge way to solving a lot of the problems in the reproducibility crisis, because then it's easy to try to reproduce analyses. And of course, a lot of this could be solved by better statistical teaching. We really need to do a better job in statistical teaching and not teaching people that statistics is some kind of cookbook that you just, plug a few buttons, and we really have to do better on teaching.
Okay, should we be particularly wary of a medical statistical study where data is discrete as opposed to continuous?
Oh, that's an interesting question. I mean, some things are discrete in life. And so not necessarily. A lot of things are measured discretely. And sometimes we take continuous variables and we make them discrete. Statisticians we would rather have the data as continuous. And we're always going to try to push you to keep it as continuous. But there are reasons, sometimes valid reasons, that you might dichotomize or break something up into categories. In medicine, we often make binary decisions, right? So we have algorithms that say if the patient has this BMI, they're obese. If we make these algorithms, and so sometimes you're doing studies that are using and testing those algorithms, and therefore it makes sense to use those categorizations. But you want to be a little bit careful about just taking continuous data for no reason and categorizing it. You want to have a good reason to do that. So you wouldn't necessarily be more suspicious. You might be suspicious of, why did the author, chop the data this way? Are they doing something funny? But yeah, sometimes things are appropriate as a discrete variable.
I think we have time for maybe one more question. So how compelling is real-world data versus controlled study data? What should we watch out for in real-world data analysis?
Okay, so I think the question is asking real-world data being just data that you would actually collect in a study versus play data that I might give you in a class. I'm not sure, or I'm not sure if they're asking about observational data versus a randomized trial. So I'll try to address both of those. So, real-world data are great because for learning in a statistical class, I will often give data sets that are a little bit cleaned up, easy to use, just for learning. When you deal with real-world data that you have gotten from a real study, there are all sorts of issues that you have to worry about: missing data, data entry errors, and things like that. So it's great practice to get a real data set to work on. So that's one thing. The difference between like observational data versus a randomized controlled trial, you can have data problems in both of those. So there's pretty good issues in both of those. You have to worry more about like controlling for confounding and things in observational studies.
All right, so I think that's all of the questions that we have time for. Again, you'll see on your screen a button to learn more about the medical statistics program and enroll in the courses. I know we've gotten a lot of questions about how to enroll, or you can go to online.stanford.edu/medical-statistics to learn more about this program. Thank you for joining us. If you found today's presentation helpful, we encourage you to share the recording with your friends, family, colleagues, or whoever might find it useful. Thank you all for taking the time to join us today, and have a great rest of your day.